Okay, been a while since I recorded one of these. This is re-recorded one from last year in an attempt to make it shorter. So this one is going to be all about the membrane, its structure, and its function. There's going to be some protein talk in here as well. So the big thing with cell membranes is that they are selectively permeable. That's a good vocab word to know, selective permeability, meaning they can kind of choose what goes in and out of the cell. Because every cell has to exchange materials with the surroundings, has to bring food and nutrients in, it has to get waste products out. And so the membrane is what kind of allows them to do this. Here is the rule for what can cross the membrane on its own with no extra help. If it is small and nonpolar, it can cross the membrane easily. That is things like oxygen gas, carbon dioxide, really small hydrocarbons. Oxygen and carbon dioxide are probably your best examples. And then large or polar molecules do not cross easily. So if it's a big molecule, doesn't cross easily. That's even something like glucose is too big to cross on its own. And glucose is actually also a little bit polar. So it will not be able to cross the membrane because it cannot get through those phospholipid tails that are nonpolar. So we're going to be talking a lot about diffusion and the different types and just different ways things can cross a membrane. And so the simplest form of this and the most basic, the easiest, is just passive transport. This is just diffusion across the membrane, no energy input at all. And this arises due to molecules always moving. The faster they move, the higher temperature they have. So since they're always moving, they're always stumbling around, bumping into each other, and this means they'll have a tendency to spread out into a space. We kind of know this. If we light a candle in one corner of the room, you can smell it at the other end of the room because the scent from that candle will diffuse through the room. So the rule is things will tend to move down a concentration gradient, going from an area of high concentration to an area of low concentration until there is an equilibrium. It's about the same on both sides, and then the movement, each direction just cancels out. So we can see it in the bottom here. At first, all the concentrations on the left, they randomly bump into each other and start to spread out to the right until they hit equilibrium, and then it's kind of equal both directions. Same thing happens when you have two solutes. They will still also tend to distribute themselves evenly. So again, down the concentration gradient. I'm going to talk a lot about concentration gradients in this. From an area of high concentration to low concentration. There's no energy needed, no ATP. Another name for this is just passive transport because it is not requiring energy. There's a special case of this. You may have noticed when I said things that can cross the cell membrane easily, I did not say water because water is polar, so water cannot cross the membrane on its own. We call diffusion of water osmosis, which is just movement of water across the membrane. And I'll talk about how water can do that in a little bit, but the general idea here with the bottom part is that if solutes can't move across the membrane, because solutes can be things like ions that can definitely not cross the membrane, that means that water will move across the membrane to try and balance the solute concentrations. This is why you can't drink salt water and feel... this is why you get dehydrated from drinking salt water, because there's more solute outside your cells than inside because the water is so salty. And so water will leave your cells to try and balance out the salt concentration outside, causing you to become more dehydrated. So what happens in these scenarios to our cells? It does depend on if you're a plant or an animal, aka if you have a cell wall or not. So we will talk about tonicity, which is basically, is this solution going to cause a cell to gain or lose water? So the prefixes here get reused and are good to know. So iso means same, and so isotonic solutions have same solute concentration inside and out, and so there's no net diffusion. Hyper means a lot of or over, too hot, like above. And this is when you have a solute concentration greater outside the cell than inside. This is the saltwater example. Your cells lose water. If you're an animal, they'll shrivel. Plant cells have a little more resilience. And then hypo means below or under. So this is solutes or solutions with solute concentrations below the concentration inside the cell, and this will cause the cell to gain water because it's going to try and balance those out. Water moves towards the area of high solute concentration, and so it rushes into the cell. In an animal, it causes the cell to burst. In a plant, it causes the cell to swell up, and this is actually its normal state. The state it wants to be in is the turgid state. Need to know these terms. Ask if you have questions on them. Okay, next. Sorry, checking. Yep. I don't know what time I have. Whatever. Okay, talking about the membrane itself, we know it is made up of phospholipids that are both hydrophobic and hydrophilic. The heads are hydrophilic, tails hydrophobic. The membrane as a whole we describe as the fluid mosaic model, where the phospholipids are kind of this fluid structure, and then you have a mosaic of proteins embedded in it that kind of move and float around within the membrane itself. A mosaic being reference to art in like tiles embedded in clay or something like that. So the membrane is fluid. It moves around. All of those little phospholipids within, the proteins within, the cholesterol molecules that you see there, they can all move and shift around, and they will be often. And so how much it can move depends on what type of phospholipid you have. And this is where our saturated, unsaturated comes in. If it's saturated, that means it is all single bonds, and it is going to be more rigid or viscous, thick, kind of like syrup, because we can really pack it in tight. Unsaturated, because of the bent tails from the double bonds, lead to more space in the membrane, allowing it to be a little bit more fluid and move around more. Cholesterol is a lipid. It's a steroid, we know, and it has a special job in our cells to help maintain the fluidity, especially in changing temperatures. So cholesterol will kind of embed itself in the membrane. It has a polar region that sticks out, and then the nonpolar rings on the inside. And its purpose is kind of just to make space and allow for things to move around. It's kind of acting just like an unsaturated phospholipid in a way. But it is good for maintaining membrane fluidity. Okay, so we've talked about things that can just diffuse across the membrane. We are now going to get into how things get across the membrane that are not small or nonpolar, which is most things. So most things will have to make use of proteins to get in or out of the cell. They will allow the hydrophilic substances, which are generally your things that are in the cytoplasm. So the first type is a channel protein, and this is just basically a tunnel for hydrophilic substances. You can really think of it as just like someone drilled a hole in the membrane, and it is a tunnel for things to go through. They are specific, though, so they don't let anything through. They will let specific molecules through. Aquaporins is the water-specific one. The way you can remember it, aqua means water. A porin is like a pore, like a pore is like a hole in your skin, right? So aquaporins are just water pores. Pores in the cell for water to enter. There are other types that I will talk about real quick. I'm not going to spend too much time on these because some of them we will get into in a future unit, but there are a bunch of proteins embedded in the membrane. So the first, transport proteins. There are various types that we will review in class, but transport proteins either passively or actively will transport solutes across the membrane. Enzymes are embedded in the membrane, but they catalyze reactions either inside or outside the cell. Don't need to worry too much about those yet. We're going to spend a lot of time with them next unit. Signal transduction is going to be all in unit 4, I believe, so I'm not going to talk too much about it, but this is just how cells talk to each other and get each other to do things that they want. And then we have cell-cell recognition, when cells can attach to each other and talk and figure out which cell is which. Again, we're going to talk about that in unit 4. Same with intercellular joining. Our cells are attached together, and also to this thing called the extracellular matrix, which is just a bunch of proteins basically that are in between cells that kind of hold everything together. Think like your collagens and things like that. So lots of proteins in the membrane. Some catalyze reactions, some transmit signals, some just serve as attachment sites, some serve as little flags saying, hey, I'm a blood cell, or hey, I'm a muscle cell. So other cells know how to interact with them. But we are going to focus on transport proteins and a few specific types. So anytime we're using a protein to run diffusion into or out of a cell, it's called facilitated diffusion because the protein is kind of facilitating the molecule's movement. They can't do it without the protein. So the first one we talked about is a channel protein. Again, just open tunnel for a specific molecule or ion to pass through. There are also carrier proteins that are not a tunnel. They're kind of like, it's kind of like an airlock where you can only have it open to one side at a time. So it's open to the outside, a molecule goes in, it causes a shape change of the protein that causes it, instead of being open to the outside, shifts to being open to the inside, and the solute then just is released and the protein resets and can do it again. So a bit slower, but more opportunity for regulation and control here with carrier proteins. Next, everything we've talked about before this is all passive. It does not require energy to do the movement. Active transport is the type of diffusion or type of diffusion, whatever, that does require energy. I remember this because if you take active transport, and remember it needs ATP, active A, transport, TP, ATP. So active transport needs ATP. It's done by specific proteins. It's going to require energy, and it's specifically going to move things against their concentration gradient. So it's going to move them from areas of low concentration to high concentration. Sorry, something was broken. So one that is good to know, because it's a common example and it kind of makes our whole body run, is the sodium-potassium pump. So this is heavily involved in all of our neurons, but will also show up in other cells. And it is a pump. And so what it's doing is it's taking energy and it's pumping sodium from the inside of the cell, where it is at a low concentration, to outside, where it's a high concentration. And then it kind of flips and reverses, and then it does the opposite, where it pumps potassium into the cell, where it's at a high concentration. So ultimately, it's creating a ton of sodium outside the cell, a ton of potassium inside the cell. If you look closely, the numbers that it actually moves is different. So it moves 3 sodium out and 2 potassium in, meaning it's creating a charge difference. It's created a voltage here, which is essentially how our neurons can send electrical signals through our body. It's run by our pumps. So much of the energy in our body, most of the ATP we make, most of the energy we consume, is used to run pumps like this one, because that is most of the work that we have to do. There are some other ways we can do this. Sometimes we will have co-transport, which is really just active transport mixed with facilitated diffusion. So it couples a pump and a transport protein. So in this case, this is a sucrose hydrogen ion co-transport. So it pumps hydrogen ions out using energy, and then as long as you have hydrogen ions and sucrose, which is just sugar, it'll move both of those into the cell at the same time. So it gives us a way to kind of regulate transport proteins, because if we turn off the pump, then the co-transporter turns off as well. So it gives us more regulation. Also, again, allows us to create a potential difference, electric potential difference of voltage. You can kind of think of it as like creating a little bit of electricity, which we can use to do other kinds of work. Okay, last bit. I know this one's a marathon. Bulk transport. So sometimes our cells say there's a lot of stuff, we need it all. So there's some special names for this. Exo and endo, remember, are prefixes that are also good to know. So exo means outer, endo means inner. So exocytosis is releasing stuff to the outside. So vesicles fuse with the membrane and then release their contents to the outside. Endocytosis is basically just the opposite. The cell membrane forms around the nutrients, pinches and forms a vesicle that then gets released inside the cell and digested, whatever. Depending on what it is, if it's eating like solid things, then it's called phagocytosis, cell eating. If it's getting like dissolved things that are dissolved in the cytoplasm, then it's pinocytosis. I've never really seen them ask specific questions on phagocytosis versus pinocytosis, but know that bulk transport does have its own special name. And it's really just using the membrane kind of like an amoeba to wrap around stuff and eat it or drink it or release it. And that, what? Didn't mean to show this. Why is it still showing that? Whoops. That's all, folks.